Chapter 16, question 8 asks, what are two different types of secondary protein structure? And to really appreciate this question, you have to have an idea of the levels of protein structure in the first place. The first level of protein structure, the primary structure, is the order of amino acids. So for example, if you had glycine, then alanine, then valine, that would give you a different primary structure than if you had alanine, valine, glycine. The difference in the order of amino acids will ultimately make a different protein. It creates the structure of the protein. It's the primary structure, the order of amino acids. In a peptide or protein. Remember, a peptide is up to around 50 amino acids, and a protein is usually bigger, more than 15 amino acids. So that's the primary structure. Secondary structure. Now, once you have this long string of amino acids, they can start wrinkling locally. So let's say you have this long string of amino acids, and then this part kind of starts to wrinkle a little bit, and this part kind of starts to wrinkle a little bit, and this part kind of starts to wrinkle a little bit. That local wrinkling in an amino acid chain is the secondary structure. Secondary structure is local wrinkling in an amino acid chain. We'll come back to that. Third, the tertiary structure. If secondary structure was local wrinkling, tertiary structure is global folding. It's the global folding of an amino acid chain. So if the original amino acid chain was just a line with like little wrinkles, the new amino acid chain will fold on a global scale. The whole thing will start to wrinkle around and make this folded thing. So it's a global folding. The entire amino acid chain folds up. That's the tertiary structure. Now, for the quaternary structure, this is the last level of structure. Proteins have 4 levels of structure. The quaternary structure is when you have multiple globally folded peptides, and actually, to make this more visible, we'll do this in different colors. This orange one is one folded peptide. This blue one is a second folded peptide, globally folded. If you have more than one peptide and they sink into each other, so they sort of come together. So they come toward each other and they clump. They don't bond to each other. They don't form any sort of covalent bond. They just clump together. That clumping is the quaternary structure. So the quaternary structure, is clumping, the clumping. That's the clumping of two or more separate, well, I should say 2 or more separate amino acid chains. The most famous example of a protein that has a quaternary structure is hemoglobin. Hemoglobin is actually 4 different amino acid chains that are not covalently bonded to each other, clumped together. Hemoglobin is the protein in your red blood cells that carries oxygen. So those are the 4 different levels of protein structure, and this question is asking about the secondary structure. So that was that local wrinkling in an amino acid chain. Now, there are several different types of wrinkles that you can form, but one is, or 2 of them actually, are really important. They are the alpha helix and the beta pleated sheet. So, with an alpha helix, the wrinkle is a sort of coil. It's like a spring or a spiral staircase. And it coils because the hydrogen on one nitrogen, remember the hydrogen is slightly positive, and the nitrogen, which is very electronegative, is slightly negative, is attracted to the oxygen of a carbonyl. That oxygen is slightly negative, the carbon is slightly positive, and so you get this attraction between them. That is a hydrogen bond. It's not a covalent bond, it's around 30 times weaker than a covalent bond, but it is the strongest intermolecular force. And so you get those repeating. They repeat and they occur throughout the molecule, and they give rise to this coiling shape. So hydrogen bonding then causes alpha helices. If you want to feel a few alpha helices right now, you can touch your hair. The proteins that make up your hair are alpha helices coiled together into bigger ropes. The other structure is a beta pleated sheet, and this is sort of like an accordion. If you were to look at a beta pleated sheet from the side, it would look like a zigzag. Or if you were to fold a paper many, many times, you would get this accordion shape and it'd be a beta pleated sheet. And you can see the same thing is holding this together. They're hydrogen bonds between the slightly positive hydrogen that's bonded to a nitrogen in the amino acid. The nitrogen is very electronegative, so it steals the electrons, leaves the hydrogen slightly positive, and a slightly negative oxygen on a carbonyl. So the slightly positive hydrogen and the slightly negative oxygen attract each other, and that holds these sheets in this sort of new formation. So again, the hydrogen bonds create this local wrinkling, this secondary structure. So the 2 most important secondary structures are alpha helices, the alpha helix, and the beta pleated sheets. An example of beta pleated sheets is the amyloid beta protein that's that's implicated in Alzheimer's disease. It's the disease actually happens when a protein that's normally in your brain, we all have it in our brain right now, cerebrospinal fluid, it just clumps up in repeating sheets of beta pleated sheets. It just clumps into a big, really tightly connected series of beta pleated sheets. And those sheets clump into big clumps, and then they stick onto your neurons and they kill your neurons and mess up all the communication in your brain. So, those are the 2 different types of secondary protein structure.